dimmi quando parti. Così.
Sei partito.
Ok. Ragazzi che siete online su YouTube, riuscite tutti a sentirci? Qualcuno ci dà un feedback sulla chat, se grossi problemi. Vabbè, comunque, buongiorno a tutti ragazzi. Io sono Filippo. Protocollo, diciamo in un certo senso, di test di ammissione per i corsi di medicina in inglese, che ha validità in un certo senso internazionale, ovvero questo programma con quesiti creati dalla Cambridge, a cui vari atenei, varie università in tutto il mondo possono unirsi per programmare l'accesso dei propri studenti nel proprio corso di medicina inglese. Perché quest'anno abbiamo attivato questa iniziativa? Perché dall'anno accademico 2019-20 anche a Padova è iniziato il corso di Medicine and Surgery, e quindi ovviamente essendo una realtà di Padova abbiamo pensato e voluto creare questa serie di La prima simulazione dovevamo farla il 9 marzo o comunque giù di lì, e ovviamente non abbiamo potuto farla per causa Covid, e siamo ripiegati a farla adesso in modalità online con il form Google, con la correzione di adesso online su YouTube. Vi pronuncio soltanto 2 o 3 cosette, nel senso che tutto il podcast della simulazione della correzione rimarrà disponibile su YouTube. Online sul sito troverete anche le slide di correzione della presentazione, e la graduatoria poi di finale con i risultati di chi ha fatto il test sul form Google entro le 10 di stamattina. L'altra cosa importante è che, essendo un corso, un test di ammissione in lingua inglese, voi avete visto che avevamo messo parte delle istruzioni, ovvero quelle più specifiche per l'evento sul sito e quelle di Eventbrite, tutte in inglese. Anche il form era tutto in inglese, poiché proprio al test sarà tutto in inglese, cioè tutta la parte di iscrizione su University, sul sito della Cambridge, e poi le varie istruzioni per trovare l'aula del test, eccetera, saranno tutte in inglese. Al test i professori o comunque i sorveglianti vi parleranno soltanto in inglese, e quindi anche noi oggi vogliamo simulare questa cosa, quindi la correzione di oggi verrà fatta tutta in inglese, con la chat di YouTube che è alla vostra destra. Interagirete soltanto in inglese e noi con voi soltanto in inglese. Questa prima parte di presentazione è in italiano, ovviamente, per permettervi di capire quello che stiamo facendo, però dall'inizio della correzione in poi sarà tutto in inglese, ok? Giusto per mettervi un attimino nel mood del test. Io non ho altro da dire, quindi lascio tranquillamente la parola a Federica, che vi spiega la prima parte dei quesiti di logica, e poi ci risentiamo alla fine. Buon lavoro a tutti. Ok, Fede, vi faccio stop share, vai pure te senza problemi.
Okay. Good morning, guys. My name is Federica. I'm first year of IMAT, and we will be doing together the first part of logic. Okay, so let's start with the first question. We had a text. As you may have seen in this in the IMAT test, very often you will find questions such as this, where you will have something to read and then to answer the question. And this is important because comprehension is a very big part of the test. So the most important thing you have to understand from this is what they are asking you. So what can be inferred from the passage? Inferring in English also means deducing. And when we say deducing, we mean extrapolating an information from the text. Therefore, it has to be something that was not explicitly stated in the text, which is why option E was not correct, because it is already said in the text. Therefore, it is not inferred. Then for the others, we can see that they are not, they are wrong because they are refuted by the text itself. So for example, referring to muscle, the term contraction doesn't imply shortening, which is not correct because as we see in the second line, the muscle may lengthen, shorten, or remain the same. So contraction in this case can also imply shortening. Then the signal to contract a muscle always originates from the brain. We know this is wrong because in the text they do tell us that there are some endogenous stimuli, for example, in the heart. And the stimulus to contract a muscle also C is wrong because, as we said, and for example, in the heart, the stimuli can be endogenous, which means that the correct answer is D. So some muscular actions can be both conscious and unconscious. This option was deduced because it is not explicitly stated in the text, and it is correct because it is what the text was trying to tell us. I hope it was easy to understand. If you have trouble understanding what I say, please tell me. I can go slower or say things again. Okay, second one. It's always a text. We saw in the statistics that many people were a bit afraid of this. We put them on the first part, and I know that it might be scary at first, but they are only 5 questions out of all of logic, and they are very important. So I do suggest revising this particular category. Still, this is about physiology and respiration. We have physiologic and cellular respiration in this text, and they ask us how they differ. If you read the text, you're going to see that each of these is correct because they take place in different environments, because one happens in the cell, while one has a whole, while one happens in the lungs and all the process that it does, the way we breathe in. Then the metabolic products, of course, because in one we have ATP, carbon dioxide, and on the other we have oxygen and water. So they differ in that way too. Of course, they differ also in the process involved because one happens in the cell and one happens in the lungs. So of course they are going to happen in different ways. And in transport of metabolites, which means all those things that are in the middle of the respiration. And of course, they are different. One uses glucose and ATP, and the other uses oxygen. So, of course, they differ in all of this, which means that E is correct. They differ in all of the previous terms. In this specific case, you could also have thought about this. If 2 were correct and 2 are explicitly stated because the environment, because we know that one happens in the cell and the other doesn't. We also know that the transport of metabolites is different. So since at least 2 of these are correct, all of them must be correct because there's no other option that includes both of them. Okay, so E is the correct answer. Okay, well, in the third text, action has to be taken now to stop the spread of bovine tuberculosis. Experts agree that reducing the number of badgers in the most heavily infected areas will help to break the cycle of infection between badgers and cattle and begin to reduce TB in both species. Although badgers can be vaccinated, there is no vaccine available to protect our cattle, and best estimates suggest that it will be 10 years before one is available. The only way to stop bovine TB spreading is to kill badgers. Which one of the following, if true, most strengthens the above argument? Okay, so we need something that strengthens this argument. And we can already exclude E because it is not contemplated. Ethically, many people do not support this solution. First of all, it does not concern anything that we said before. And secondly, in any case, it will not strengthen this case. Then, when efforts are made, the D also does not strengthen, it weakens the argument. Also C, because in both cases, this action of killing badgers is not enough to prevent TB in cattle. Then B also, because if we say that vaccinating would be the better option, we are not strengthening. So B, C, and D weaken the argument. So the only one that so the only one that is correct is A, because in this case we see that in order to vaccinate a badger, we will have to take 5 shots, which is something that will be time-consuming and also expensive, probably. So the best way to get rid of the problem is to get rid of the badgers and not to prevent the TB, because it would take too much time, and that would not necessarily make things better for the cattle in the meanwhile. I hope you understood. Okay, see there are no questions. Okay, 60% of the people registered in a sports center do swimming and 70% play tennis. The total people registered in the sports center are 200. How many of them do both swimming and tennis? Well, this was a mathematical problem. And in this case, of course, you first had to calculate how many people do swimming and how many do tennis. So you just had to do the 60% of 200, which is 120, and the 70% of 200, which is 140. Now from this, what you had to do is take either of them and subtract them from the total number of people. So it was 200, for example. Let's do the people that are swimming. So 200 minus 120, which makes 80. You could also have done 200 minus 140. That was a viable option too. But for now, we're making an example using the people that are swimming. So the total is 80. We then subtract the amount of people participating in tennis by the amount of students not participating in swimming. So 140, which is the number of people that we know are doing tennis, minus those people that are not doing swimming, which is 80. And the total is 60. And this is the amount of people that for sure is doing both. You could also have done the opposite, so 200 minus 140, and what you had, take it away from 120, which was 60. So 200 minus 140 was 60, then you would have done 120 minus 60, and the result is always 60. So the correct answer is E. A 400-gram packet of snacks has the following nutritional values. It contains 10.86 grams of protein per 100 gram and 1.5 grams of protein per snack. How many snacks does the package contain? Well, in this case, the best thing is to use a proportion. So we do 10.86 to 100 is equal to 1.5 equals to X, which means that if we now turn this around, so to find the x, we will find x equals 13.8 grams per snack. Then dividing the total per the result that we just got, we have a package that contains 29 snacks in total. So, okay. I hope this is clear. Okay. Two runners, Liz and Laurie, are training for a competition. Rory passes the start point every 9 minutes. Liz passes the start point every 15 minutes. They have just set off on their routes. How long will it be before they meet up at the start point again? In this case, you have to find the minimum least, the least common multiple, quindi il minimo comune multiplo in italiano, quindi di 9 e di 15. Poi il risultato, then the result, you had to divide it and breaking it down to prime factors, and the result is 35 minutes. As you know, to find the least common multiple, you just have to multiply 9 by 15, and the result breaking it down in prime factors. So do the 2 columns until you arrive to 45. So the correct answer is A. I wish to, now number 7. I wish to tile an area of a wall 240 centimeters wide by 200 centimeters high. Tiles are 20 centimeters square. I will therefore need that 12 per 10 equals 120 tiles. Which of the following uses the same methods of calculation as above? Please do notice that in this case, we do not need to calculate anything. We just need to find which of these follows the same reasoning. Don't be scared by all the numbers. You need to identify the pattern and find it in one of the options, which in this case was D, because we have the length of the sides of the cube, which in this case would work both for the height and also the width, because our cube, of course, has the same length on each side. And then would they also tell us how many we would need to fill a certain space. And therefore the reasoning is the same. And therefore the correct answer is D. This one for me was one of the hardest because you had to read correctly this scheme. The figure below shows the result of a survey of 1,000 people based on the size of the current and previous car. So how many of them would you expect to trade up to a larger car than the present one? Okay, in this case, what you had to do is try to understand, see which of them would trade for something bigger, which means that you take in consideration the people that right now have a medium-sized car and previously had a small-sized car. Let's find the two. Let's find these people in the scheme, which are the people here. So 200 and 200 plus this one. So the people that previously had a small car, because you see previous and small, 80, and current and medium, which is 200. These are the people that are most likely to next get a larger car, because previously they showed to be interested in buying first a small car and then a medium car. So we would assume that next they will buy a large car. Now we know that they are in total 280. So now what we do is 280 divided by, we need to find the percentage over 1,000 people. So of course we do 28 over 100 multiplied by 1,000, and the result is 28%, which means the correct answer is D. please pay attention. It was a bit hard to read the scheme. Keep in mind you have to find previous car as the small one, which is 80, and current medium car, which is 200. Add them together, so 280, and go from there. Very often you will find questions as such. Okay, number 9. A jeweler sells a watch with an 18% reduction on the price. That is a $36 reduction. What was the price of the watch? Well, in this case, we would call the initial price. The best way to do this, the fastest way, was to call the initial price X. So the 18% of X is 36. Therefore, equals is equal to 36 multiplied by 100 over 18. And if we calculate this, the result will be 200. And so we found the initial price. So the correct answer is D. Okay, last one. In order to succeed in academic examination, it is necessary to study. Therefore, if a student works hard in a particular subject, he or she should do well when the time of the examination comes. Which of the following best describes the flow in the argument? Okay. In this case, we see that D is the correct answer. It assumes that studying harder is a sufficient condition for the academic success. Among all the other ones, we see that this one is the most likely, because it ignores the fact that some subjects are more academic than others. We don't have a definition for which subjects are academic or not. We do not decide that. Then B, it overestimates the value of studying in preparation of examinations. Not really, it just says that people who study are going to do well. Then it assumes that it is necessary to study in order to succeed. This is not a flaw in the argument. This is something that is true. It ignores the fact that some students do not need to study very much in order to succeed. We do not know that. We cannot assume that. The best flow we can find is D. It assumes that studying hard is a sufficient condition for academic success, which we know it is not, because people might have problems. This is a common thing. Sometimes studying is not enough for academic success. And among all of these, this was the one that could be proven right, while all the other ones could be subjective. This one isn't. Because just to say, we could say that for academic success, first you need to apply to a certain school. And that's an objective thing that changes the outcome, while all the other ones can be subjective. So the correct answer is D. Okay, we did a lot of logic together. Now we'll pass to my colleague, Matilda, who will do general knowledge with you. I hope you understood. Wait, Nagur asked to watch again the number 8. Sure thing. Yes, of course. It's a bit of an hard question because you had to read correctly this scheme. So in this case, please see that we have previous is on this side. So we calculate, so every previous is on this one. So the current one is everything that goes down. So the columns. And previous are the lines. Now we need to see who of these people is more probable to get a larger car. The people that are more probable to get a larger car is the ones that first had a small car and then currently have a medium car. So we have a 200, which is current medium, 200, and previous small, which is 80. We do 200 plus 80, which is 280. And from that, we calculated the percentage, which is 200, which is 28 multiplied by 100% multiplied per 1000. And the total is 28%. I hope you understood. It's just a matter of reading this correctly and understanding what the question wants. You need to understand who is going to get a larger car, and these are the two options. We're going to avoid the large part because these do not matter to us. We're going to avoid the people that currently have a small car because apparently they do not care for something bigger. Otherwise, first they would have gotten a smaller one, and now they would have a medium. Just need to calculate the people that had first a small car, then got a medium car, and so we can assume that next will have a large car. So again, 80 plus 200. Okay, now I'm done. I will pass to Matilde. Thank you very much for listening, and have a good continuation.
Hi everybody, I'm Matilda. I'm attending the first year of medicine and surgery, and I'm gonna correct the first 6 questions of general knowledge. So let's see the first one. Which one of the following was the author to a series of short stories named Dubliners? So, as the question says, Dubliners is a series of short stories which talks about the relationship of, indeed, Dubliners and their city. This is a work written by James Joyce, who is one of the most influential writers of the 20th century. We can see that the other options are all writers except for Turner, who was a romantic painter. Baudelaire was a French poet who's known for his poem collection called The Flowers of Evil, and Kipling is famous for The Jungle Book. So the right answer is C. Who produced and detected X-rays in 1895? So the right answer is A. Marconi was known for his radio transmission works. Hilbert was a mathematician, and Marie Curie was a famous French chemist who also won a Nobel Prize. What happened on the 3rd of March of 1918? So, in this date, Russia decided to no longer participate in the World War I because it had some internal issues. Because indeed, in 1918, the Russian Civil War began. So the right answer is B, since Russia signed with the other Central Powers the Treaty of Brest-Litovsk. In which of the following states there is an higher percentage of Christian than other religious groups? The right answer is Ethiopia, so letter E. We see that in Sudan, the main religion is Islam. In Pakistan is Buddhism. In China, we have many different religions, such as Buddhism, Taoism, and Confucianism. And in North Korea, there is no main religious because North Korea is an official atheist state and it has no religious holidays. In which years was the independence of United States declared? So the United States became independent on the July the 4th of 1776. And as you can know, the 4th of July is the national holiday in the United States. So here's my last question. What's the capital city of New Zealand? Nowadays, the capital is Wellington, but the first one was Old Russell, and then the second one was Auckland. But since 1865, the capital of New Zealand is Wellington. So my work is done, and I'm gonna leave you with Lucrezia. Thank you. Bye.
Okay, hi everybody, I'm Lucrezia. I'm a first year student of medicine and surgery, and now I'm gonna correct the last 6 questions of general knowledge. So the first question was, when was the Magna Carta Libertatum documented, agreed to? The Magna Carta Libertatum, called also Magna Carta, was a charter of rights that was signed in June 15 of 1915 by King John of England. The main goal was to make peace between the unpopular king and a group of rebel barons, and he promised the protection of church rights as well as the protection of barons against the illegal imprisonment, the swift of justice, and the limitation of the feudal payments to the crown to be implemented through a council of 25 barons. So the correct answer is the C, in 1215. So which of the following branches of the European Union was Ursula von der Leyen as president? Ursula von der Leyen is a German politician. She is the president of the European Commission since December 1st of 2019. So the correct answer is the A. She served in the federal government as the longest serving member for Angela Merkel from 2005 until 2019. And she's currently the member of the center-right Christian Democratic Union, as well as its European counterpart, the European People Party. The 19th question is, who is the author of the Rime of Ancient Mariner? The author of the Rime of Ancient Mariner is S.T. Coleridge. So Samuel Taylor Coleridge is an English poet, and the Rime of Ancient Mariner was written in the years in between the 1797 and the 1798. It was published in the first edition of the Lyrical Ballads in 1798. So in ancient times, the pigment purpura was obtained from, the correct answer is A type of shellfish. The purpura was obtained from a peculiar type of shellfish, which is the murex. In the past, it allows the Phoenicians to make a fortune out of it because they were the only ones to know how to obtain this precious and really desired color. So Luis Sepulveda is a famous writer that, among other novels, wrote the story of the seagull and the cat who taught her to fly. He was born in, the correct answer is D, Chile. He was born in Ovalle, Chile in 1949, and he recently died due to the coronavirus in Oviedo, Spain in April 16 of 2020, after he was diagnosed as the first man with the virus in the region of Asturias in Spain. The type of government that runs in China at the moment is, the correct answer is D, People's Republic, governed by the Communist Party. China is, as well known as People's Republic of China, PRC, is governed by the Communist Party of China with Xi Jinping as a president. It is currently one of the few existing socialist states, as well as one of the world's only socialist state that is explicitly aiming to build communism. So these were the questions of general knowledge. If you have any questions, just ask them in the chat, and my work here is done. Now we're gonna correct the questions of biology.
Good morning, guys. I am Vincenzo. I am in my 3rd year of medicine and surgery, and I'm gonna correct your questions of biology. So let's move forward with the first one. Wait, here. The cellular matrix provides support and connects the cells, is also known as Glycocalyx, is secreted by animal cells and many bacteria, is present only in plant cells. Well, here, you know that it does provide support and connects the cells, and it's secreted by animal cells and many bacteria. As for the Glycocalyx matter, well, Glycocalyx is the part of the cell outside that bears glycoproteins that are used, for example, in inflammation and stuff like that. So it is part of the matrix. I am not sure if it is a synonym, but if you have to choose the rightest one, you have to go for this, because you know that it's not only present in plant cells, and these two are true. So the answer is B. 1, 2, and 3 are the correct answers. Which of the following statements about DNA is false? Well, wait, just inform me that the slides are not very well seen. Well, I'm gonna, wait, I'm gonna try and okay. Just give me a minute so I can try to fix this. Is it better now, guys? I'm gonna go for the old school. Okay, perfect. So let's move on. It's not that fancy, but I guess the content is what matters. Which of the following statements about DNA is false? It is a polymer to polynucleotides and stuff. Let's move on to the explanation. Well, what we know is that it is a double helix, and what we know that is actually false is that two polynucleotide strands would run each other in a double helix, but they are not held together by covalent bonds because they would be too strong, and in order to be replicated, it has to be opened. So it is actually a pretty strong bond, but it's made up from the sum of singular hydrogen bonds, which you know are weak bonds. But this is something that my colleagues from chemistry are going to explain you well. So the correct answer is, in fact, B, as it is a polymer. It involves 4 different bases. It was Rosalind Franklin's work that was not recognized at the times, but she did help, and it contains a pentose sugar, nitrogen-containing bases, and phosphate groups. Moving on, which of the following processes are catabolic reactions? Well, what's catabolism? Catabolism means breaking down, basically. So what do you do when you break down? Well, you know that protein synthesis is not at all a breaking down. Actually, it's the opposite. And so is photosynthesis. I am not very good at vegetative cell metabolism because we don't study that in medicine, but you have to know that. So I guess you know better than me that photosynthesis is making up glucose out of carbon. So instead, you know, the glycolysis, you know, glucose, which is sweet, and lysis, luo, melt. And so it is a catabolic reaction, and so is fermentation, which is anaerobic, but still consists as a breaking down molecules. When you have in front of you words difficult as catabolism, anabolism, which is the opposite, try to think about the etymology in order to get to the meaning of that. So correct answer is 2 and 4, because glycolysis and fermentation are the only processes that are actually a breaking down of stuff. 26, in case of hyperglycemia, an excessive amount of glucose circulates in the blood plasma in order to bring blood sugar back to normal. Well, there are a lot of things. This is something that might sound unintuitive, but if you think about it, you have a lot of sugar in the blood and you have to take it in the cell. It's still in the body, but it's not in the blood anymore, so it's better. Therefore, what happens in a very difficult pathway that I'm not going to explain you now because you'll have a lot of time to study that in your university, the insulin stimulates the uptake of glucose so that it's inside the cell and it's not outside, it's not in blood. It's better. Even because it's the entry is stimulated in the right cells, so such as the liver, that does the exact opposite of what is said in the A answer, which is converting glucose to glycogen so that it can be stored, or it's converted to triglycerides and then stored as in adipocytes and stuff like that. So the correct answer is C. You stimulate the entry of glucose inside of the cell so that it's not in the blood anymore and it doesn't go around making a mess. 27, in which of the following cells the number of chromosomes is equal or less than it's duplicated. I'm sorry, I didn't notice that. A female haploid cell. Hepatocytes, erythrocytes, and sperm. Well, that's kind of easy. You can think, which of the following is the closest cell to an ovocyte? Well, I would say sperm, because they are both, involved in reproduction. So they are haploids, so that they can meet, they love each other so much, and they make new babies. While erythrocytes don't have a nucleus, so they are, I don't know if that's a word for that, but they have no chromosomes at all. While hepatocytes are diploid cells, they are normal autosomic cells, and they don't need to have less cells than usual. So you know that the correct answer is D, that only sperm. You have to think about the meiosis also. That is a process that involves only the spermatocytes and ovocytes. So the answer is C. This one is a bit long. It's gonna take me a lot of time to read this, but let's go. So 4 medical students, Clara, Giacomo, Valentina, and Filippo, recorded different structures that they thought were found in a healthy animal red blood cell, a liver cell, and a typical maple leaf cell. So we're clearly talking about 2 animal cells and a vegetal one. So they stated the structures, and now we're gonna go for each one. So Clara found centrioles in red blood cells. She found maple ADS ribosomes in maple leaf cell and well-defined core in liver cell. Well, in red blood cells, I don't think you have centrioles, but anyway, she is not right, of course, because yeah, they don't have most of organelles, apparently that's the word, and so that they can accommodate maximum space for hemoglobin. Yeah, moving on to Giacomo. Well, he says the red blood cell has hemoglobin, right? And as for maple leaf cell, where ADS ribosome, it's a bit complicated because you have to know the specific isoform of ribosomes that you have in eukaryotic and prokaryotic cells. As for prokaryotic cells, they have this 70S ribosome, and animals, they have the 80S. So in maple leaf cell, you don't find 70S ribosomes. That's a bit tricky, but if you learn that, you're gonna go. And so Giacomo is not right either. While Valentina, well, she found DNA strands in red blood cells. She's wrong because we don't have any DNA inside of the red blood cell. That's something that I said in the answer before. So that's basically it. Also, we don't have cell wall, of course, in liver cell. And Filippo, well, he found hemoglobin in red blood cells. He's right. Plasmodesmata, again, I am not a specialist of vegetal cells metabolism, but they told me apparently that there is a plasmodesmata in maple leaf cells. And they found mitochondria in liver cell, which is actually very important for their metabolism. So, in conclusion, we know that the answer is C because Filippo is the only person who is right because the others found structures that weren't there actually. In case you have any questions, of course, don't bother to write on the live YouTube streaming. They are telling me currently what's going on. So if you need me to explain something to you again, I would very much do that. 29, which of the following hereditary diseases is autosomal recessive? Albinism, achondroplasia, or dwarfism, Huntington disease, sickle cell anemia. Well, actually, albinism has 2 isoforms, but actually, none of them is autosomical dominant. Achondroplasia or dwarfism is autosomical dominant. Huntington disease is 2, and sickle cell anemia is recessive as well. So you have a very long explanation here, but basically what you have to remember is that albinism, Huntington disease, sickle cell anemia, and achondroplasia, they're specific transmission way. And you know that albinism and sickle cell anemia are both autosomal recessive, while achondroplasia and Huntington disease are both dominant. So that the answer here is E, that only 1 and 4 are the right ones. I mean, autosomical recessive. 30, the first metabolic intermediate that is common to the aerobic metabolism of glucose and fatty acids is acetyl-CoA, beta-hydroxybutyrate, pyruvate, citrate, glyceraldate, 3-phosphate. Well, you know that beta-hydroxybutyrate, it's actually a ketone body that is common only to fatty acids way. Pyruvate is only of glucose. Citrate, it's actually, I think, in between. I don't have that much memories of my biochemistry exam, but it's not the first one. Glyceraldate 3-phosphate, it's part of the glycolysis process. I think it's the 5th reaction. I don't actually remember that. But we know that the first one is acetyl-CoA. The answer is A then, because acetyl-CoA is carboxylated in order to make malonyl-CoA, which is the first molecule in the process of lipogenesis. So it's the first common metabolite, also because in the opposite, the beta oxidation brings to formation of several molecules of acetyl-CoA. And you know that we get acetyl-CoA from glucose metabolism when we dehydrate pyruvate. So the right answer is A, acetyl-CoA, as it is the first common metabolite. The Duchenne muscular dystrophy is caused by a mutation of the dystrophin gene located on the short arm of the X chromosome. Which of the following statements are false? Females typically are carriers of the genetic trait, while males are affected. The daughter of a carrier mother has a 50% chance of being a carrier. The DMD is a degenerative neuromuscular pathology. The son of a carrier mother has a 50% of inheriting the defective gene from his mother, and the son has a 50% chance of inheriting the disease from his father. Well, I would like to go straight to the right answer here, which is actually the false answer, is E, because, the son is a son in case he receives the Y chromosome from his father. In case the father is affected because he has an X chromosome. And if he had the X chromosome, surprise, you're not a son, you're a daughter. So you have no chance of inheriting the disease from a father. As for the rest, well, DMD is a degenerative neuromuscular pathology. This kind of information I really like because you have to know that. It has to be part of your general knowledge. And you can't logically derive that. the daughter of a carrier mother has a 50% of being a carrier. That's true. Females are typically carriers. It's very difficult for a female to develop the X-linked diseases because they have to have both chromosomes mutated, and it's very difficult for that to happen. While the son of a carrier mother has a 50% chance of inheriting the defective gene is actually the same as the daughter, only basically he has 50% of inheriting the defected chromosome. So we have to move on. I'm sorry that you see me as a ghost because I have my window behind me, but probably it's better like that. Which of the following molecules contain only carbon, hydrogen, and oxygen? Cellulose, hemoglobin, amylase, triglyceride, and DNA. Well, let's go for that. Cellulose, you know, it's a polymer of carbon. It's a polymer of glucose, not exactly glucose, but anyway, glucides. So you know that it has only carbon, oxygen, and hydrogen. Hemoglobin, amylase, they are both proteins, and as proteins, they bear aminic groups, so they do have another molecule. Triglycerides, well, again, this is a fatty acid, and as you know, fatty acids only bear carbon, hydrogen, and oxygen, while DNA not only has aminic groups in the structure of the bases, I think it's the word, but it also has phosphate groups. So the right answer here is A, which is 1 and 4 only, because as I said, hemoglobin and amylase are both proteins, and DNA is actually a lot of atoms stuck together. 33, the Krebs cycle is carried out in the mitochondrial matrix of eukaryotic cells, in the mitochondrial matrix of prokaryotic cells, in the cytoplasm of eukaryotic cells, in the cytoplasm of prokaryotic cells. Well, this one is pretty easy. It's also kind of unintuitive, but trust me, you have to know this kind of stuff. We know that the Krebs cycle is carried out in the mitochondrial matrix of eukaryotic cells. That's easy, actually, you know that. You also know that prokaryotic cells don't have mitochondria because mitochondria are probably remains of prokaryotic cells themselves. So you don't have them. And therefore you can exclude both these ones because, you know, they are mutually exclusive. The fourth one, in the cytoplasm of prokaryotic cells. Well, beginning with the fact that by exclusion, and as I said for the first question, the only one that includes, that doesn't include a wrong answer and includes the right answer is the one featuring 1 and 4, so the C. Well, the cytoplasm of prokaryotic cells, I didn't know that before, I have to be honest, but apparently the Krebs cycle is carried out also in the cytoplasm of prokaryotic cells. What I knew is that prokaryotic cells do have ATP synthesis, you know, so they have mitochondrial, no, not mitochondrial, but of course the chain of electrons. Except is in the membrane, the only difference between the ATPase from the mitochondria and the one from prokaryotic cells is that the ATP synthase is in the prokaryotic cells has more subunits, so it takes more time to spin around and form ATP, so more protons are needed in order to form an ATP molecule. This is something just for your curiosity. And yeah, so the correct answer is C. Again, here, in case you didn't know this, and trust me, there is going to be a lot of stuff that either you don't know or you feel like you don't know, you can deduce that from the fact that C answer is the only one that has a right answer and doesn't feature a wrong answer for sure. So you can go for C. I guess this is the last question. No, we still have 2. Which of the following options are true about mitochondrial genetic diseases? ATP metabolism may be affected. That's true. Mitochondria, as one of the main purposes, is to make energy. Famous catchphrase, mitochondria is the powerhouse of the cell. Actually, it's not, because they don't produce energy, but they convert it. They can be inherited by male sons. They can be in case they are from a mother. They can be inherited by female daughters, the same as one before. They can be inherited from sick fathers. That's true, actually. And they can be inherited from sick mothers. They are true. That's true too. It's a pretty tricky question. I think the person that wrote this was pretty much of a joker, but enigmist. But you know that the answer is D. Something that I want to tell you is that during your exam, you're going to be very anxious, and questions like this are going to sound like, I don't know how to say, an enigma. So please breathe deeply and read it very well, because it's easy to be tricked in this case. But if you read it correctly, it's a pretty much easy question. So the correct answer is D, because we saw that, well, we saw that before. They do have ATP metabolism affected, can be inherited from sick fathers, they can be inherited from sick mothers. DNA replication, and this is the last one, guys, is fully conservative, is absent in cells infected with viruses, happens in the first phase of mitosis, involves different enzymes such as transferases, leads to different DNA strands. Well, DNA replication is not fully conservative, it's actually semi-conservative. I think it's the right word also in English. Is absent in cells infected with viruses. I'm not gonna go specifically speaking about viruses, because you'll have a lot of time to speak about that. But you know that viruses, sometimes the lysogenetic ones especially, they induce the replication. So this is not correct. Happens in the first phase of mitosis. Well, no, because the first phase of mitosis is prophase. It's not the wrong one. I'm gonna go to the last one because it's not correct either. Leads to different DNA strands. Well, although you have some small mutations, you cannot say that they are different DNA strands. In general, we assume that the cell is so good at replicating that these DNA strands are exactly the same. So this is not correct either. Involves different enzymes such as transferases. Well, I didn't know that. Another word for I think transferase is the helicases. I don't know. Well, yeah, RNA polymerase. I'm sorry. It's a particular transferase. I didn't remember that. I hope you do remember that. And although, well, you could have guessed that by exclusion, because you know that all of these are wrong, and it does involve different enzymes. So we're gonna trust on that and say, well, perhaps one of these is a transferase. So guys, this is all for me. Ask me whatever you want on the YouTube live. And I'm gonna leave the word to my friends from chemistry, and bye y'all, have a nice day and good luck with your test.
Good morning. Here was Rob. There's anatomy before chemistry. And so we'll go on correcting anatomy if everything is all right. So let's begin. So human anatomy and physiology just after biology. Just after biology. So let's go on. Okay. Which of the following structure is or are part of the electrical conduction system of the heart? SA node, sino-HL node, AV node, His bundle, or Purkinje cells? And then there are the answer, as always, Cambridge style. So let's go on and discuss the electrical conduction system of the heart. First, the impulse that regulates the contraction of the heart generates in some special cells in the sino-HL node or SA node. Then it's conducted via three preferential ways, like big motorways, to the atrioventricular node. Then it propagates through the east bundle and through the east and to the left and right branch of the conduction system of the heart, and at the end at the Purkinje fibers that stimulates the contraction of the ventricular walls. So let's go on and discuss the of the heart. So the SA node located in the right atrium, it's sinoatrial because it's in the atrium, stimulate the atria to contract. Then the impulse goes to the AV node where it stops there, so the atria can contract and push all the blood through the valves into the ventricles. There, after this small delay, the signal diverges and is conducted through the east bundle, and then on the left and right branch, and then on the Purkinje fibers, causing the electrification and the depolarization of all the muscle in the ventriculi and the contraction of the ventricular chambers. So these signals are generated rhythmically, about 60 to 80 beats per minute in the ASA node. So there's a rhythm which can be changed, but normally it's a rhythm and it has nothing to do with the brain. So the heart has its own way of generating beats and rhythm. And this signal generates the coordinated rhythmic contraction and relaxation of the heart. This signal, electric signal, can also be read in the ACG or AKG, it depends on where you are from. And you see the waves are the electrical signal and the electrical impulses. So Purkinje cells, which were the last options, has nothing to do with the heart, are neurons, which are in the cerebellum, are GABAergic because they use that neurotransmitter, and they're located in the cerebellum where they help to regulate balance. So again, let's discuss the alternatives. SA node is part of the electrical conduction system of the heart and is where the impulse is generated. AV node is where the impulse stops, so the atria can contract, push all the blood through the valves into the ventricles, and the signal stops, and then the signal goes through the east bundle and then into the Purkinje fibers to give the impulse to all the muscle in the ventriculi. Purkinje cells are neurons, so they are not part of the electrical conduction system of the heart. So the correct answer was B, 1, 2, and 3. 37. Of course, if you have any questions, just write them on the YouTube chat and I will answer them later. Choose the right answer. The osteoclasts have the same cytologic origin as the osteoblasts, are a type of osteoblast that's following an excess in bone production at a quiescent state, produce and secrete the unmineralized organic portion of the bone matrix called the osteoid, and are usually more active than the osteoblast, or come from the same precursors as the macrophages. So let's discuss the osteoclasts, what they are. As you know, probably, osteoclasts are one of the 3 kinds of cells that compose the bone, which is the organ that composes the skeleton. The osteoclast is the bone cell that breaks down bone tissue. This there's another kind of cell, which is very important, that builds up bone tissue, that secretes the matrix, which is the osteoblast. So osteoblast secretes the matrix and builds the bone. Osteoclast destroys it. It destroys by eating, because it comes from the macrophagy. So it's an evolution of the macrophagy, differentiation of the macrophagy, which becomes the osteoclast. There's another kind of cell, which are osteocytes, which are inside the bone matrix, and they develop when the osteoblast has secreted too much matrix, that it's enclosed into the matrix, so it can't secrete anymore because the cell is enclosed in mineralized matrix, and so it becomes an osteocyte, which is a quiescent cell, which doesn't secrete and doesn't destroy, does nothing. It helps with feeding the bone and controlling it, but the shoot cells that build and destroy osteoblast and osteoclast. So let's go on through all the options. So the osteoclasts have the same cytologic origin as the osteoblast, as we said no, because they come from the macrophagy. Macrophagy, we will discuss later, are a kind of blood cell. They come from the bone marrow. We will discuss later. Are a type of osteoblast that follow an excess in bone production and hematopoietic state? No. The cell that answer B refers to are osteocytes. Then they produce and secrete the demineralized organic portion of the bone matrix called the osteoid. No. Osteoblast. And they're usually more active than the osteoblast. No. This is important. There's a balance. They're usually as active always. Bone is not a fixed organ. It's a continually moving and shaping, evolving organ. So the osteoblasts build and osteoclasts destroy. Usually there's a balance, but there is a very important disease which may happen also in the IMAT test, which is osteoporosis. That happens when osteoclasts are more active than the osteoblasts, so more matrix is destroyed than built, and so the bones are more fragile and more susceptible. Are more fragile, and this leads to easier fracture happen. So they come from the same precursors as the macrophages as we read before. Of course, again, if you have any questions, just write them down. Which of the following statements about the adrenal gland are correct? It's located beneath and slightly median to the kidney. It is divided in two sections, the cortex and the medulla. The medulla secretes stress hormones like cortisol, or the cortex can also secrete aldosterone in response to the stimuli of the renin-angiotensin-aldosterone system, which is the RAS system. And then always like the Cambridge-style combination of answers. So adrenal glands. Adrenal glands are two endocrine glands which are located above the kidney. They have a slight different size and shape depending on the side. The right one is leafy-like. It's like a leaf. And the left one is a pyramid. So you can, they're different. And they're divided in two parts, both histologically because they have a different cellular composition, and embryologically because they have a different embryological origin. They come from different embryonal tissue. So the adrenal cortex is gland derived. The embryological origin is a gland. It's a pure gland. And it's conventionally divided in three zones. From the outside to the inside, we have zona glomerulosa, which produces aldosterone in response to the angiotensin II, produced by the RAS system, which regulates blood pressure. We will discuss it later, just a bit. And then there's zona fasciculata, which produces cortisol in response to stress. And zona reticularis produces androgens, in particular DEA, under hypophysis stimulus. So zona glomerulosa, zona fasciculata, you see all the fasciculi, all the vertical lines, and zona reticularis because the blood vessels make like a net. The medulla produces adrenaline or epinephrine. It depends on whether you're from the US or you speak American English or normal English, and noradrenaline, which is norepinephrine, in response to sympathetic stimulus. The difference is it's neural driven, so it's like a modified nerve. It's modified nervous ganglion. And let's go. We go on with the explaining what are the stimuli that make these glands secrete. So first, adrenal medulla, which is the innermost part, which is nerve derived, secretes adrenaline and noradrenaline in response to sympathetic stimulation. So sympathetic stimulation, nerve signal, gives the adrenal medulla the sign to produce adrenaline and noradrenaline, which are proteins, very important. Two hormones, but they are proteins. In contrast to all the hormones secreted from the adrenal cortex, which are steroids or more like fat-like hormones. So zona fasciculata and reticularis secrete the innermost parts, secrete androgens and cortisol in response to pituitary signals and hypophysis stimulus. There is this important axis, this important endocrine axis, which is the hypothalamic pituitary adrenal axis. So the hypothalamus tells the hypophysis to secrete ACTH, which leads to the synthesis of androgens and cortisol. Cortisol in response to stress, and androgens to also androgens in response to stress and other stimuli. And it has a negative feedback. So if there is too much cortisol, then the hypophysis can feel there is too much cortisol and stops producing ACTH. This is normal negative feedback. Then zona glomerulosa, which is the most difficult part, which is the outermost part, produces aldosterone in response to low blood pressure. So low blood pressure means low renal perfusion, which means a sympathetic activation, and some special cell, the juxtaglomerular cells of the kidney, produce renin, which produces after a series of reaction angiotensin II, which leads zona glomerulosa to produce aldosterone. What does aldosterone do? It mainly lets you keep in sodium and water, so you increase the volume of the blood and thus increase the blood pressure. It's a bit difficult, but it's like this is just a spot of things you have to know when you are ready for the test. If you have any question, again, just go in the YouTube chat. So the following statement about the adrenal gland are correct. 2 and 4. It's divided into sections, the cortex and the medulla, and the cortex can also secrete aldosterone in response to the stimuli of the renin, angiotensin, aldosterone system. 1 is wrong because it's not located beneath, but it's above the kidney. And the medulla secretes stress hormones, yes, but not like cortisol. Cortisol is a steroid, like epinephrine and norepinephrine, which are proteins. Again, number 39. Which white blood cell are elevated during allergic reactions? Eosinophils, basophils, neutrophils, or lymphocytes? We're discussing about blood. Blood is composed of plasma, which is the liquid part of the blood that contains water mostly, and many proteins, which are very important. Albumin, coagulation factor, and some other proteins which are very important. We'll discuss later. And cells that are produced in the hematopoietic organs, bone marrow mostly, from the hematopoietic stem cell. In the fetus, a very important hematopoietic organ is the liver. But again, in the adult, the normal, when you are in the adult, also in the child, the main hematopoietic organ is bone marrow. There are 2 lines of development of blood cells. The lymphoid line, from the hematopoietic stem cell comes the lymphocytes, which are these cells. And the myeloid line produces everything else, which is erythrocytes or red blood cells, monocytes, eosinophils, basophils, neutrophils, and platelets, which come from a big cell, which is a megakaryocyte. White blood cells are counted in the complete blood count and can be differentiated in kinds. Usually, white blood cells are 4 to 11,000 per milliliter of blood. And the scale of frequency, there are neutrophils, which are 50 to 70% of total white blood cells. Lymphocytes are 20 to 40%. Monocytes are 3 to 10%. Eosinophils are 3%. Basophils 1%. You can remember this useful phrase, never let monkeys eat banana. So never has 5 letters. So you have about 50% neutrophils. Let has 3 letters, so about 30% lymphocytes. Monkeys, you have 7 letters, about 7% monocytes. Eat, 3 letters, 3% eosinophils, and bananas. Then if you don't let monkeys have any banana, they have 0% banana, so you have 0% basophils. It's an easy mnemonic phrase, very useful to remember not only the order but also the quantity of the kind of blood cell in the complete blood count. Then white blood cells can be divided in 2 main categories, which is polymorphonuclear granulocytes and mononuclear agranulocytes. Polymorphonuclear means the nucleus has many shapes. Actually, it has many lobes. And are granulocytes because they have granules in the inside which can secrete substances. Mononuclear, they have only one nucleus, are only monolobe nucleus. Are granulocyte because they have no granules to secrete outside. The most important polymorphonuclear granulocyte are the neutrophils, which are big cells with multi-lobe nucleus. They are increased in number in bacterial infections, and they are professional phagocytes. They eat the bacteria. Eosinophils are big cell with multi-lobe nucleus, and they are called eosinophils because they stain red due to the color of their granules. They are increased in number in parasitic infection, mainly worms and allergic reaction. They are so important in parasitic infections that you can find them usually, not pathologically, in the gut. You find them, it's not uncommon to find them there. Then basophils are big cells with a multi-lobe nucleus, 2 or 3 lobi, and they stain it blue. That's why they call basophils, due to the color of their granules. They contain a receptor for histamine, which is the main mediator with IgA, immunoglobulins, of allergic reaction, where they are increased in number. As you see, neutrophils, eosinophils, and basophils take the name from their affinity or to the stain, which is used to color the blood, which is Giemsa usually, or hematoxylin and eosin. They're neutrophils if they stain normal, eosinophils if they stain red, more eosin, and basophils if they stain blue from hematoxylin. Just a funny thing to know and to remember what they are. Mononuclear granulocytes are mostly monocytes, which are big cells with a single nucleus, and they're differentiating macrophages in the target tissue. As before, they're differentiating macrophages in target tissue or osteoclasts in the bone, which is a particular kind of macrophage, and they're increased in number in the late stage of infections. So, and lymphocytes are small cells with a single nucleus that fills the whole cell, and they're increased in viral infection. So, what to remember? Neutrophils, bacterial infection, and they're increased in number in the first stages, early stages of infections, together usually with eosinophils and basophils, because these ones, the polymorphonuclear granulocytes, are the cells that fight, firstly, the bacteria that invades the body. Then, when the battle is over and the immune system has won, you have to clean up. To clean up, you need macrophages, which clean up all the mess that were invaded into the battle. And macrophages derive from monocytes. That's why you find them in late stages of infection. And then lymphocytes are increased in viral infection because virus attacks the cell, enters into it, so you cannot destroy it without destroying the cell, and the lymphocyte destroys the cell which was infected with the virus. Very easy. The question was about allergic reaction. Allergic reaction is a kind of immune response. It's very easy actually. When you first come in contact with an allergen, some people which are more sensitive than others to that allergen become sensitized. So the allergen, which is this response, stimulates some molecules to produce IgE immunoglobulins, which bind to basophils, you see them, and also to mast cells, mostly basophils. And they're there, and basophils remain there with this IgE trigger to response when the allergen is presented again. So when the allergen comes in again, the second time into the body, IgE recognize it, they bind to the IgE in the basophils, which then presents it to the mast cell that secretes histamine immediately. Histamine is the main mediator of the first stage of allergy and causes the main reactions that are important in the allergic reaction, also in anaphylactic shock, which is, is the most dangerous kind of allergy. And then eosinophil, together with basophil, start secreting some other cytokines and other substances that produce some substances such as leukotriene, very important in the late stage of infection of allergic reactions. So what is increased during allergic reactions? Mostly eosinophils and basophils. Basophils because they bind to the allergen and say to the mast cell that they have to secrete histamine, and eosinophil because together with the basophil they have to secrete the late substances of anaphylaxis or of allergic reactions. Number 30, last question. Which of the following statements about the liver is or are correct? The liver is an organ which detoxifies various metabolites. The liver is an organ which synthesizes proteins. The liver is an organ which produces biochemicals necessary for digestion and growth, and is located in the right upper quadrant of the abdomen below the diaphragm. So let's go on. What is the liver? The liver is a big gland, the biggest gland of the body, which is both endocrine and exocrine. It produces substances important from the inside of the body, such as hormones, insulin-like growth factor 1 is the most important one, and exocrine to help digestion and to help such as bile. So you see, the liver is located below the diaphragm in the right upper quadrant of the abdomen. Here, you can see. It's supplied by the hepatic artery that gives oxygen to the liver, and there's a peculiar vascular system, which is the portal circulation. The portal circulation means that the portal vein, which comes from the gut, brings all the nutrients absorbed into the intestine into the liver by dividing many branches and many small vases. Here, the portal veins enters into the hepatic lobule and brings all the nutrients to the hepatocyte that metabolizes them. If there is some nasty substance such as alcohol, they detoxify it, or also some drugs such as paracetamol or acetaminophen. It depends on where you come from. Acetaminophen is tachipirina in Italy. And they detoxify it, and then the blood, with all the nutrients taken away, is thrown into the central vein, which then goes through the hepatic vein into the inferior vena cava, and then on to the heart and the rest of the body. So nutrients, when are absorbed into the gut, do not go all the way through the body, and then they enter the liver later through the artery, but they enter the liver through the portal vein. The liver produces bile, which is a greenish substance that helps to digest lipids. Bile is excreted into the duodenum via the bile ducts. When you have excess bile, it's stored into the gallbladder, which then squeezes when you need it to digest food. So the liver is composed of many of these liver lobules, which they have a portal vein on the outside. The blood goes into these small vases, which are these sinusoids. They're called sinusoid because they have a snake-like appearance, and they are fenestrated, which means they have many windows and opening that let all the nutrients flow free outside of the vase. The nutrients are all then metabolized and stuff into the hepatocytes, which surround the sinusoid, and then all the blood goes into the central vein to go then to the right heart and the rest of the body. There's a very important kind of cell in the sinusoid, which is the Kupffer cell, which is another of the macrophages of the tissue macrophages. So you know, monocyte differentiate into macrophagy in the tissue. The macrophagy of the liver are Kupffer cells. What does the liver do? The liver metabolizes all nutrients and many toxic substances, such as alcohol, and as I said before, acetaminophen. It synthesizes many proteins, such as albumin, which is the most important protein of the blood, which gives blood its tone. blood is not fluid like water, because it has albumin that keeps some pressure in it. Then coagulation factors, which help clotting of the blood if needed. Complement factor, which are very important for the immune response. Then haptoglobin, ceruloplasmin, transferrin, protease inhibitors, which are all important in the early stages of infections. The liver is very important to immune response because it produces many factors which are involved in inflammatory reaction. And then the liver is an essential storage of iron, copper, and vitamins A, D, and B12. They have a different storage capacity. For vitamins A, for example, there's a storage of many years if someone has a good diet. Vitamin D is stored, but it's not as much storage space as vitamin A. Vitamin B12, there's about 1 or 2 years worth of storage. That's why the most important side effect of vegan diet is B12 deficiency, because B12 is exclusively produced by animals. And so B12 deficiency does not happen immediately, doesn't happen in a month or two. It happens in about a year or two. So it's very difficult to understand it at first, but it causes many problems. And then it excretes bile salts and bilirubin, which are exocrine substances, goes into the duodenum. And then some important hormones, as I said before, the most important one is insulin-like growth factor 1, which you can find also as EGF1, which helps to use sugars and insulin, and it grows some part of the body. It helps to use sugars and nutrients of the body. So, again, we will discuss the statements. The liver is an organ which detoxifies various metabolites. Yes. It detoxifies all the normal metabolites which you assume in the body with the food. So, glucose, proteins, amino acids, and stuff. But also nasty substances, such as alcohol. Then the liver is an organ which synthesizes protein. Yes. The most important one, the one you have to remember, are coagulation factors and albumin. And then the liver is an organ which produces biochemicals necessary for digestion and growth. Yes. Digestion is, for example, bile, and growth IGF-1. And it is located in the right upper quadrant of the abdomen, below the diaphragm. So they are all correct. I have ended. I finished my presentation. If you have any questions, just write them. I will go and try to answer them if there are any. Thank you for your attention. If everything was, I know maybe something was not very clear, but so there are some things which are very important to remember. If everything is all right, just okay. Okay. I think everything is okay. So thank you, and I'll give the word to chemistry. Have a nice day.
Hello, hi everyone. I'm Giovanni, third year of medical school. So we can start with the chemistry part, question number 41. Okay, so in an experiment, 10 cubic centimeters of carbon monoxide and 10 cubic centimeters of oxygen are mixed. The gases react as shown in the equation below. So what will be the total volume of gas present in cubic centimeters at the end of the reaction? And remember that all the gas volumes are measured at the same temperature and pressure. So the question is the total volume of gas. So first of all, you need to balance the equation. So 2 moles of carbon monoxide plus oxygen gives 2 moles of CO2. So since all the gases are kept under the same conditions for pressure and temperature, the mole ratio that exists between the species involved in the reaction becomes equivalent to the volume ratio. So you can either use the moles or the volume to solve the problem. So since we have a ratio between CO and O2 of 2:1, the reaction will consume a volume of CO twice as large as the volume of oxygen. Carbon monoxide will act for this reason as the limited reagent because it will be consumed before all the oxygen gets a chance to react. So given a ratio of 2:1, we have 10 cubic centimeters of CO. And for this reason, we have only 5 cubic centimeters of oxygen reacting in the reaction. For the same reason of a ratio of 2:2 between the reagent and the products, we will have 2 moles of CO2 produced in the reaction. So a total of 10 cubic centimeters of CO2 at the end of the reaction. So remember, the question was how many was the total volume of the gas present. So we have the 5 remaining cubic centimeters of CO that didn't react in the reagents, and the 5 cubic centimeters of CO2 that were formed in the reaction. So at the end, we will be 5 plus 10, total 15 cubic centimeters of gases in the reaction. Next question. Okay, this was a little tricky. What is the freezing point depression of a solution of 0.16 moles of sodium carbonate in 100 milliliters of ethanol? Okay, so to calculate the freezing point depression, you have to use this formula that involves molality. Molality is the different thing from molarity, which we used to use, and it is the number of moles of solute over the weight of the solvent in kilograms. And remember that the freezing point depression depends on the number of particles present in the solution. So for this reason, we have to use the Van 't Hoff coefficient. So that for sodium carbonate is 3, because in a solvent, sodium carbonate dissociates in 2 sodium ions and 1 carbonate ion. So to solve the exercise, we have to find the mass of the solvent, knowing its density and its volume. And remember that density equals mass over the volume. So the density of ethanol is 789 kilograms square meters over square meters, which basically means, if you do the conversion, 789 times 10 to the minus 4 kilograms over milliliters. The volume of the solution was 100 milliliters, and the mass was 0.0789 kilograms. So if you replace all these numbers in the formula on the top of the slide, you can find the freezing point depression of 12. So the correct answer is actually D. Next question. Right, which of the following is or not redox reaction? So the first one, phosphorus acid gives phosphoric acid plus phosphine. If you can go to the correction slide, okay. This is a disproportionation reaction. So the phosphoric acid acts both as the oxidizing agent and the reducing agent. So the phosphorus acid gives phosphoric acid and phosphine. And if you pay attention, the phosphorus change is oxidizing number. From +3, it goes to +5, so this is the oxidation, and it goes also to -3, this is the reaction. So this is a redox. The second reaction is phosphoric acid that gives copper oxide to give copper phosphate and water. So this is not a redox reaction because the oxidation numbers are the same. So the phosphorus in the reagent is +5, and the phosphorus in the products is +5, and the same is for the copper, +2 and +2. So we don't have electrons moving in the reaction, so this is not a redox one. In the third one, we have potassium permanganate, where the manganate change is oxidizing. Manganese change is oxidizing number from +7 to +2, so it reduces its oxidizing number. And the iodine that goes from -1 in the iodine to 0. So this is the oxidation reaction. So this is a redox reaction. So the only one which was not a redox reaction was actually the B. So the second reaction. So the correct answer is 2 only. Many elements, question number 44. Many elements in nature can have different isotypes. So although one isotope commonly predominates due to stability, the other isotypes can affect the overall mass of the element. If we suppose a certain element has 2 main isotypes, isotope A has a molar mass of 128, and isotype B has a molar mass of 132. 1 mole of this element, when found in nature naturally, weighs 128.8 grams. What are the percentage of isotope A and B when found in nature? So to solve this problem, you have to make an hypothesis. So if these 2 isotypes were equal in nature, 50-50, then a mole of the element in nature would weigh 130 grams. And to have this number, you have to make the following math. So 0.5 times 128 grams plus 0.5 times 132 grams gives 130 grams. So, and this is for a 50-50 ratio. Since the number is less than we can conclude that isotype A is more dominant than isotype B, lowering the total average mass. So for this reason, we can exclude answers A, C, and E. So now the isotype A will make up a certain percentage of the element in nature. We can call this percentage X, while isotype B will make up the rest of the percentage. So 1 minus X. If you do the math, 128X plus 132 plus 1 minus X equal 128 plus 0.8. This will give an X of 0.8. So 80% of the element is composed of isotope A, and the other 20% is composed of isotope B. So the correct answer is D. So 80% and 20%. Last question for my part. So which of the following configurations represents the correct and most stable structure of ammonia and phosphine, so phosphorate trihydride? But it's not the last question, sorry. The chemical model used to determine the correct structure of every molecule is the VSI-PR theory, so the valence shell electron pair repulsion theory. The most important principle of this theory is that the valence electron pairs surrounding an atom tend to repel each other and will therefore adopt an arrangement that minimizes all the repulsions. In our case, we have 2 molecules, which are ammonia and phosphine. So nitrogen and phosphor belong to the same group, so the 5th group of the periodic table, and present an electron pair on top and 3 unpaired electrons which constantly repel each other. So the electron pair tends to push away the 3 hydrogen atoms, thus blocking a possible tetrahedral structure. So the correct answer is actually the D. So AX3E, it's a trigonal pyramidal structure for ammonia and phosphine. So question number 46. Okay, which of the elements listed has the following electron configuration? So 1s2, 2s2, 2p6, and 3s2. So the options are, okay, don't worry. The question can be resolved simply by counting the number of electrons and remembering the position of the various elements on the periodic table. So 1s2, 2s2, 2p6, 3s2. So remember that an s orbital has 2 electron tops and a p orbital has 6 electron tops. So if we can calculate the total number of electrons by adding all the electrons in the different orbitals together. So 2 in the first one, 2 in the second one, 6 in the third one, and 2 in the fourth one. So 12 total electrons. For all the atoms that are ions, we know the total number of protons is equal to the number of electrons. But in this case, if we see aluminum has 13 protons, but since it's an ion, it has a negative charge, and the total number of electrons is 14 and not 13. So the response E is incorrect. Manganese has 25 electrons, so response A is for some reason incorrect. Chlorine has 17 electrons, and response C is again incorrect. Selenium has 34 electrons, and response D is incorrect. The only one with the correct number of electrons is magnesium, which has 12 electrons. So the correct answer is A. I'm done, and I'm gonna hand it over to Mario for the last six questions of chemistry. If you have any questions, just feel free to write it down on the live. I'm gonna answer them all. Thanks, everyone.
Hello, can everybody hear me?
Yes.
I'm having some trouble with opening Zoom. Oh well, in worst case, we can just go with the audio. Anyway, everybody, let us start.
You're good, Mario. You can go.
I'm good. All right, I'll just go then. Oh wait, never mind. I found it, guys. Seems good. All right. So everybody, nice to see you. My name is Mario. I'm a first-year medical student, and I'll be doing the second section of the chemistry section of the exam. So let's start with number 47. Which one of the following molecules is a structural isomer of cycloheptane? So A, heptine, B, 2,4-dimethyl-2-pentene, C, methyl-cyclohexane, D, benzene, and E, 2-ethylpentane. So if we go to the next slide, we can see that the structural isomer is a type of isomer in which molecules with the same molecular formula have different bonding patterns and atomic organization. Cycloheptane, which is this one right here, has a molecular formula of carbon 7 and 14 hydrogen. And so the point of the question is to search the other molecules with the same molecular formula. As we can see, heptine has 7 carbon and 12 hydrogen, so that obviously can work. Methyl cyclohexane has 7 carbon and 10 hydrogen, benzene 6 carbon and 6 hydrogen, and 2-ethylpentane has 7 carbon and 16 hydrogen. Meaning the only possible solution, which is 2,4-dimethyl-2-pentene, this one right here, is the B. So moving on to number 48. Consider the redox reaction. Sodium iodide plus 3 molecules of hypochlorous acid creates sodium iodate and 3 molecules of hydrochloric acid. So which of the following statements are correct? Number 1, chlorine gains electrons. Number 2, sodium iodide is the oxidizing agent, or number 3, the oxidation number of iodine is +5 in the sodium iodide. So, a quick review. In a reduction oxidation reaction, oxidation is the loss of electrons or an increase in the oxidation number of an atom. Reduction is the gain of electrons or a decrease in the oxidation number of an atom. The oxidizing agent reduces itself, while the reducing agent oxidizes itself. The oxidation numbers in the given reaction are as follows. And looking at this, we can clearly see that iodine oxidizes itself, going from an oxidation number of -1 to +5. So sodium iodide is a reducing agent. And chlorine reduces itself, as we can see, it goes from a +1 to a -1. So hypochlorous acid is the oxidizing agent. So that means that while 3 is correct, it isn't the only correct answer. B is incorrect because sodium iodide is not the oxidizing agent. That is, in fact, hypochlorous acid. And D is incorrect for the same reason as B, and E is incorrect for the same reason as B. And so the only possible solution is C, which has both number 1 and number 3, excluding number 2, which is incorrect. Going on to number 49, a solution of nitric acid contains 0.0 moles of nitric acid in 100 milliliters of pure water. If you dilute 10 milliliters of this solution in enough water to make up a solution of 1 liter, what is the pH of the obtained solution? So, going on, we're gonna have to remember the formula for molarity. So to calculate the molarity of the initial solution, we have to remember that molarity is equal to the number of moles of the solute divided by the total volume of the solution. And doing this, we obtain 0.0 moles, doing simply 0.01 molar, doing simply 0.01 moles divided by 0.1 liters, which gives us 0.1 molars. The number of moles contained in 10 milliliters is the product between the molarity and the volume, which is simply 0.1 molars times 0.01 liters, which gives us 0.001 moles. We can now calculate the molarity of the second solution with the previous formula, obtaining 0.001 molars, doing simply 0.001 divided by 1, since the total volume is 1 liter. Knowing that the nitric acid is a strong monoprotic acid, the concentration of hydrogen ions is equal to the molarity of the solution, and therefore we can calculate the pH using the formula. pH is equal to negative logarithm of the concentration of hydrogen ions, which in this case is 10 to the negative 3, which gives us the solution 3. And the correct answer then is E. Going on with number 50, which of the following molecules can have a cis-trans isomerism? Number 1, but-2-ene. Number 2, butene dioic acid, or number 3, 2-butyne. So, as you might know, cis-trans isomerism, also known as geometric isomerism or configurational isomerism, is a type of stereoisomer. Cis indicates that the functional groups are on the same side of the carbon chain, while trans conveys that the functional groups are on opposing sides of the carbon chain. As you can see here, the most important substituent is methyl, being the only substituent. Here you can see that this is a cis configuration, this is a trans. Most of the time, the molecule must have a double bonding in order to have this isomerism. So 2-butyne can't have cis-trans isomerism, since it has a triple bond and not double bond. By drawing the other two molecules, you can see that they both have a cis-trans isomerism. Here representing the butene, and here representing the butene-dioic acid. You can see where it turns. And so that means that the correct, oh, so there's another slide, my bad. Oh, right. The cis-trans isomerism is a spatial isomer, also known as stereoisomer, as mentioned previously, which is different from constitutional isomers, which are also known as structural isomers, since that simply has a difference in where the element is located. This actually has movement in space, which is an important difference. Knowing this, we know that the solution is A, since number 3 is wrong because, as we said earlier, triple bond for the 2-butyne. Number C is also wrong for the same reason, since it includes the number 3. Now, D is incorrect because while the 2 is correct, both the 1 and the 2 is correct. E is wrong for the same reason as B and C, and so the only solution is A. Going on to number 51, 5 of the ions of vanadium are listed below, as you guys can see. Which 2 ions of vanadium are in the same oxidation state? So, in all the molecules in the question where oxygen is present, its oxidation number is -2 in the question. Knowing this and the total charge of the molecules, we can calculate the oxidation number of vanadium for all the molecules, using a simple formula to calculate the oxidation number, giving the variable x, of vanadium in the first choice. So, essentially, we know that the total charge is -1. And we know that the oxidation number of oxygen is -2. And we also know that there's three oxygens in that molecule. So giving x a variable for vanadium, we do +3 times -2, which is equal to -1. Using the simple equation, we can see that the oxidation number of vanadium is +5. The oxidation number of monoatomic ions, like in number 2 and 3, equals the charge of the ion. So the oxidation number of the second and third choices is +3 and +2 respectively, as you can simply see here. For 4 and 5, using a similar formula we used for the first choice, we can find the oxidation number for vanadium in the 4th and 5th choices. So using the formula, we can see that in the 4th choice, the oxidation number of vanadium is 0. And in the 5th choice, we can see that the vanadium has an oxidation number of +5. With this, we can see that 1 and 3 is wrong, since that has an oxidation number of 5 and 3 has an oxidation number of 2. 2 and 5 is wrong, since 2 has an oxidation number of 3 and 5 has an oxidation number of 5. 3 and 4 is wrong, since 3 has an oxidation number of 2, 4 has an oxidation number of 0. And E is wrong, since 4 vanadium has an oxidation number of 0 and 5 vanadium has an oxidation number of 5. Meaning the only solution is C, where both the vanadiums have an oxidation number of +5. Moving on to number 52, which is actually the last question for chemistry. The acetyl salicylic acid, ASA, also known as aspirin, which is how we'll call it from now on since that's a complex word, is an organic compound belonging to the NSAID family, also known as non-steroidal anti-inflammatory drug family, which contains two important chemical functions. Which options identify these functions? So you can see here that aspirin contains both ester, right here, and the carboxylic acid here to the left, which are the following structures. Looking at the image provided to us here, we can see that the carboxylic acid, you can see my cursor, should be on the top, and the ester is to the right. Knowing this, the solution to number 52 is E, and you can easily see it. And that should be it for chemistry. After us should be math and physics. So we're done here. I'll pass it over to them. I hope you understood everything correctly. And without further ado, I'll pass you to them.
So, hi everybody. My name is Angelica. I'm from first year and I will correct math. So, the first question was, which of the following expressions is equal to this equation? The solution to this equation was first to write each number as a power of 3, which means we recognize 9 as 3 to the power of 2. And we must remember to add its own power from before. So it becomes 3 to the power of 2 to the power of 2n. The same way, 27 is 3 to the power of 3 and its power n. So we have 3 to the power of n to the power of 3 to the power of n. And at the denominator, we just keep 3 to the power of n. Now, it is important to remember the property of the powers, which means that a power raised to another power is a power with the same base and the power which is the product of its exponents. So we will have 3 to the power of 2 to the power of 2n, which means having 3 to the power of 4n, and on this time 3 to the power of n times 3. We can now simplify 3n with 3n, and so we'll have 3 to the power of 4n times 3 to the power of 2n. And now we know that when we have two numbers, a power of two numbers with the same base and different exponents, we just have to do the sum of the exponents. So we'll have 3 to the power of 6n, which means the right answer was letter B. Now, number 54. John bought a car for 18,000 pounds. After 1 year, the car loses 10% of its original value. The second year, it loses further 15%. So what is the price of the car after these 2 years? So we have the initial price, which was 18,000. We have to calculate the first devaluation, which was of 10%. This means we can operate in two ways. The first is by timing the original price by the complementary of 10%, which means 90 over 100. And so we get 16,200. Otherwise, we can calculate with 10%, which means 18,000 times 10 over 100, which gives us 1,800. So now, doing the initial price, which was 18,000, minus the price we've just received, which is 1,800, we'll get 16,200, which is the same number as we did before. For the second devaluation, we must be careful because we don't need to do it on the original price, but on the price we just calculated, which means doing the complementary of 15, which is 85. So we'll have 16,200 times 85 over 100, and we'll get 13,770. And the answer so was letter E. The diameter of the wheel of a motorcycle is 13 centimeters. How many revolutions will it make to travel 99 kilometers? So, we must consider the wheel as a circle of diameter 63 centimeters. We can calculate the circumference of the wheel of the motorcycle, which is phi times diameter, and we'll get 188 centimeters. The total distance was given in kilometers, so we must pay attention and convert kilometers in centimeters. So we'll times 1,000 to get to meters, and then another times 100 to get to centimeters. We must imagine now our wheel as a straight line. So to calculate the number of revolutions, we might imagine there is a distance and our wheel, which is another distance in the biggest distance of 99 kilometers. So we'll have the number we just found, converting 99 kilometers in centimeters, over our circumference of the wheel, which was 198. And this gives us 50,000, which is the answer to our question. So 50,000 revolutions. The last question for maths was, in the given figure, lines x, y, x, y, so this, and mn, intersects at O. If POY forms a 90 degrees angle and the ratio between A and B is 2/3, we must find C. So at first we can name A and B as A as 2 times x and B equals 3 times x. So from the picture, we can recognize xy is a straight line. Raise O and M and OP stands on it. So xy is a straight line, OP and OM, OP and OM stands on it. So we know that the sum of B plus A plus this right angle gives us 180 degrees. So we write A plus B plus 90 degrees equals 180 degrees. We now write A as 2 times x and B times 3 times x. So we have 2x plus 3x plus 90 degrees equals 180. We can now solve this equation and we find that x was 18 degrees. Then calculating A, we have 2 times 18, 36, and B, 3 times x, which is 54. But the question asks us about C. So we must go and see, since MN is a straight line and OX stands on it. So we have OM and then OX. And we know that B plus C is another 180 degrees. So we can now write as before, B plus C equals 180 degrees. So we then write B as 54 degrees plus C equals 180 degrees. C then, resolving the equation, becomes 126 degrees, which gives us answer letter B. This is a part of maths, so if you have any question, please write on the YouTube channel. Thank you.
Hi everyone, I hope you can hear me. I'll introduce myself. I am Andrea and I am from the first year of medicine and surgery. I'm now gonna correct the last 4 questions of the test, the physics part. Okay, so starting from question number 57, the two cylinders of a hydraulic press filled with an incompressible fluid have a radius of, respectively, R and 10R. If a force of 120 newtons is applied to the piston of a smaller cylinder, what is the acceleration of a car with a mass of 1,000 kilograms placed on the plate of the bigger cylinder, considering the gravitational acceleration being 10? So in order to solve this question, you had to know a basic principle of the physics of fluids, and that is Pascal's law. Now, Pascal's law simply states that when a pressure is applied on fluid, it spreads, it propagates to the whole fluid. So the pressure that I apply in a point of the fluid, I will also find in every single other point of the fluid, if the fluid, of course, is incompressible, like in the problem. So the pressure that I am applying with my force of 120 newtons on the smaller cylinder, I will also find on the bigger cylinder where the car is placed. We know that we can calculate the magnitude of this pressure just by dividing the force by the area. And we know that this will be equal to the pressure on the bigger cylinder, which also will be equal to the force, this time the force that the fluid applies on the car, divided by the area of the bigger cylinder. And so we can write the equation that you see here, FA over SA equals FB over SB. We know FA because it's the force that we are applying. We know SA because we know the radius of the smaller cylinder. We know SB because we know the radius of the bigger cylinder. And so we can calculate FB, inverting the formula. And so FB will be equal to FA times SB over SA, which equals 120 newtons, which is our force, times 100 pi times r squared. And then we don't really need to know the radius because then we divide this by pi times r squared, which is the area of the smaller cylinder. And so pi and r squared simplify, and we're left with 12,000 newtons, which is the force that the piston, the fluid, will apply on the car. But then, of course, we know that the car has a mass, so it'll have a weight force itself. To find the resulting force, we have to, because the two forces are in opposite directions, we have to take the force that the fluid is applying on the car and subtract the weight force of the car, which of course will be equal to the mass of the car times 10, because the gravitational force, as you know, is G times the mass. So what we're left with is the resulting force, which is 12,000 newtons minus 10,000 newtons. And in order to find the acceleration, we have to divide this by the mass of the car, which is 1,000 kilograms. And so resolving this equation leaves us with 2 meters over second squared, which was the acceleration of the car. And so the correct answer was D. Moving on to question number 58. Of course, if you have questions, leave them in the YouTube chat and we'll be answering them. Question number 58. A capacitor with an initial capacitance of C equals C0 or CO is filled by a fourth with a dielectric medium of relative permittivity epsilon equals 4. What is the final capacitance of the device? Now, you're probably familiar with problems regarding capacitors, but this is a bit of a peculiar problem because we have a capacitor that is only partially filled with the dielectric medium. So there's a simple rule that we can apply to solve this problem. And the rule states that if a capacitor is filled only partially with the dielectric, we can consider it to be equal to a system of 2 separate capacitors connected in series, one corresponding to the capacitor that has the dielectric, and it'll have a distance between the plates of only a fourth in this case, because only a fourth of the initial capacitor was filled with the dielectric. And then another capacitor will be equal to the part of the initial capacitor that doesn't have the dielectric. And so it'll have a distance between the plates of 3/4 the initial distance. Now we can use the classic formula to calculate the capacitance of the capacitors, given the area of the plates, the distance between the plates, and the relative permittivity, starting from C1, which is the capacitance of the smaller capacitor with the resistor with the dielectric medium. So the relative permittivity will be 4. The area is the standard area, and the distance between the plates, as previously said, will be a fourth of the initial distance. So it'll be d over 4, and so this 4 goes up, and eventually we're left with 4 times 4 times CO equals 16 CO. So this is the capacitance of the capacitor with the dielectric. Then the capacitor that doesn't have the dielectric, as previously said, will have a distance between the plates of 3/4 the initial distance. It doesn't have the dielectric, so there's no factor that we need to multiply epsilon to. And so we're left with a capacitance of 4/3 times CO, of course, because we know that CO is simply epsilon times S over D. So because the two capacitors are connected in series, we have to use the formula of the two capacitors in series, which is the one below here. So 1 over the equivalent capacitance equals 1 over the capacitance of the first capacitor plus 1 over the capacitance of the second capacitor. Inverting the formula, we have that the equivalent capacitance is equal to the capacitance of the first times the capacitance of the second over the sum of the two. And so substituting C1 and C2 with the ones that we previously found, eventually we're left with 16 over 13 CO. So that was the final capacitance of the system. And so the correct answer was B. And moving on to the question number 59. This one was a bit easier, I guess, or at least it was a bit quicker to understand, but you had to know a bit of theory, because the question was, which one of the following is not a fundamental physical quantity? And the options were electric current, temperature, time, electric charge, and luminous intensity. Now, of course, you had to know what a fundamental physical quantity is. And it is simply a quantity that is considered to be fundamental. Okay, so there's a series of physical quantities that are fundamental, and all the others are derived from these initial physical quantities. So fundamental physical quantities are the basis of any dimensional analysis. And the international system deems 7 physical quantities to be fundamental. And they are, well, some of these are pretty obvious, like time, mass, or temperature. There's also length. But others might be a bit more tricky, like electric current, luminous intensity, which you probably haven't heard of, and amount of substance, which you probably actually heard of in chemistry. So looking at the different options that you had, and knowing what a fundamental physical quantity is, you could already understand that the correct option had to be one between electric current and electric charge. Because if one of these two is a fundamental physical quantity, then the other is not, because one can be derived from the other just by multiplying or dividing by time, as you know. And I'm pretty sure a lot of you thought that the electric charge was fundamental, because intuitively it seems more fundamental, but the international system actually considers the electric current to be fundamental because, for historical reasons, the electric current has been discovered and measured before the electric charge. And so the electric charge and its unit of measure, the coulomb, was measured multiplying the electric current by time. And so the correct answer was again D, because electric charge was not and is not a fundamental physical quantity. And so I'm going to move on to the last question of the test, which also was probably the toughest question of physics, and it required a little bit of knowledge of operations between vectors. So given two vectors, R and S, with the length of R being different from 0 and the length of S is being different from 0, which of the following options is or are necessarily, pay attention to this word, necessarily correct? Option number 1, R cross product S is perpendicular to R. Option number 2, R plus S is perpendicular to S. And option number 3, R plus S dot product R cross product S equals 0. Now let's analyze, and then you have all the different options according to the Cambridge style. Let's analyze all the 3 options, starting from the first one. So that little X that you see between the R and the S is a symbol for an operation, a common operation between vectors, which is the cross product. And it's easy to remember because it's cross. And cross product is has a symbol that is a cross. And the cross product between two vectors is also called the vector product because the result is a third vector that is necessarily perpendicular to the 2 initial vectors. The magnitude of this vector can be calculated with the formula that you see here. So the length of A, that is the cross product of R and S, will be equal to the length of R times the length of S times the sine of the angle between the 2 vectors. So you can also actually understand what will be the direction of the resulting vector using the famous rule of the right hand. But to solve this problem, you didn't have to know that. You only had to know that the cross product results into a vector that is perpendicular to the plane on which the initial vectors lay. And so the first option had to be correct, is necessarily correct. Okay. And then let's move on to the second option. Maybe this was a bit easier because we're all familiar with the sum between vectors. At least I hope so. So R plus S is perpendicular to S. As you probably know, you can calculate the sum of two vectors using the rule of the point tail, which is explained by the image that you see here. So the magnitude of the resulting vector, the sum between two vectors results into, of course, another vector that has a magnitude that depends both on the length and the angle between the 2 initial vectors. So it could be true, the option could be true, the sum could result in a third vector that is perpendicular to one of the initials, but it's not necessarily true because we're not provided with the angle between the initial vectors. And so the second option was not necessarily true. And this is why you had to be careful about that necessarily term in the question. And let's move on to the third option, which kind of mixed the first and the second. The sum between R and S, dot product, that little dot that you see there, the cross product between R and S equals 0. So let's start from the sum that we have already discussed, ends up in a vector that is that of course will lay on the same plane as S and R. And in the image, it is indicated with the letter B. And then the cross product, whereas the cross product will result into another vector, here indicated with the letter A, that necessarily has to be perpendicular to the 2 initial vectors. So the sum is B, and the cross product is A. And these 2 vectors, A and B, have to be perpendicular. Now, what is that little dot that you see there? It is a dot product which is another very important operation that you can make between vectors. It is indicated with a dot, but it is also called a scalar product because the result is a scalar, it's not a vector. And the scalar can be calculated using this formula. So A dot product B is equal to the length of A times the length of B times the cosine, not the sine this time, the cosine of the angle between the two vectors. And so because A and B are perpendicular, the angle between them is 90 degrees. And as you know, the cosine of 90 degrees is 0. Hence, this dot product will result into a scalar that is 0. And so the third option was correct. So the correct answer was D, because only the first and the third option were necessarily correct. And this will be it for physics and for the whole test. And I guess now I'll leave the word to Federica for the final greetings. Thanks for sticking around to this point. And if you have questions, of course, just let us know.
Thank you very much. Okay, guys, so I'll give you the last greetings. Posso parlare in italiano adesso per dare gli ultimi saluti. E Andrea e Filippo, che è il nostro coordinatore, doveva andare via, quindi vi dico le ultime cose importanti. Intanto la graduatoria è già online, non sono state contate i compiti i test delle persone che l'hanno lasciato in bianco. Comunque è stato il primo test in inglese, quindi non importa, il prossimo sarà a luglio e sicuramente questa è stata una prima prova anche per noi. Ricordatevi poi che il prossimo test sarà quello di italiano il 23 maggio. Per domande o qualsiasi altra cosa, per informazioni, ci trovate sui social, su Facebook e Instagram, mi raccomando seguiteci. Detto questo, ho detto tutto e vi ringrazio per essere venuti a questa correzione e a presto.